Hello and welcome to your ultimate guide for the D313 OA, Anatomy and Physiology course. My goal today is simple: give you everything you need to fully understand the key concepts, master them, and walk into your D313 OA exam with total confidence. In this video we'll cover digestion overview, gastrointestinal histology, digestive anatomy, enzymatic absorption, neuroendocrine regulation. With over 100 plus success stories, we have proven our videos will help you pass your exam on the first attempt. And remember, our website offers free exam guides and practice questions to help you ace your D313 OA exam. You'll find the link in the description below, so be sure to check it out and get the resources you need to study smarter, not harder. So let's get started.
Let us start with the big picture. The digestive system has one job: to turn food into molecules small enough for your cells to absorb. Your cells cannot import a piece of apple. They take in tiny building blocks like glucose, amino acids, and fatty acids, so food must be broken down first. The system has two parts. First is the alimentary canal, a continuous muscular tube about 9 meters long, running from mouth to anus. Food actually passes through it. In order, it includes the mouth, the pharynx, the esophagus, the stomach, the small intestine, and the large intestine before reaching the anus. The second part is the accessory organs, which aid digestion but never have food pass through them. These include the teeth and tongue, 3 pairs of salivary glands, the liver, the gallbladder, and the pancreas. If you swallowed a marble, it would travel the whole canal, but never enter the liver or pancreas. Digestion happens in 4 key stages, and your exam loves their order. First is ingestion, simply eating. Second is digestion, the mechanical and chemical breakdown of food. Third is absorption, where small molecules pass into your blood and lymph. And fourth is defecation, the elimination of whatever cannot be used. That second stage has 2 flavors you must keep straight. Mechanical digestion physically breaks food into smaller pieces without changing its chemistry, like chewing and churning. Chemical digestion uses enzymes and acid to break the actual bonds holding molecules together. Both work together since smaller pieces give enzymes far more surface to attack. One more idea ties this together: propulsion. The movement of food along the canal. The start is peristalsis, a wave of muscle contraction squeezing food forward like toothpaste from a tube. For your exam, remember the phrase, "I would do anything for donuts," giving you ingestion, digestion, absorption, and defecation in order. Now let us zoom into the wall of the gut tube, because almost everywhere it has the same 4 layers. Knowing them from inside out is a classic exam question. Picture 4 coats around the hollow tube. From the lumen outward, they are the mucosa, the submucosa, the muscularis externa, and the serosa. The innermost layer is the mucosa, which touches food directly and handles secretion and absorption. It has an epithelial lining, connective tissue beneath, and a thin muscle layer. The lining changes by location. At the mouth, esophagus, and anus, it is tough stratified squamous tissue, while the stomach and intestines use simple columnar tissue. Just outside lies the submucosa, loose connective tissue packed with blood vessels, lymphatic vessels, and nerves. It is wonderfully stretchy. When you eat a big meal, this elastic layer expands to let the stomach fill, then recoils when it empties. It also carries the nerve plexus that controls local secretion and blood flow. The third layer is the muscularis externa, responsible for movement. Usually it has 2 smooth muscle sheets, an inner circular layer that narrows the tube, and an outer longitudinal layer that shortens it. Together they create peristalsis. Here is a high-yield fact. The stomach adds a 3rd oblique layer, making its wall the thickest. The outermost layer is the serosa, a slick membrane that reduces friction so organs slide smoothly, and it anchors the tube in place. In the esophagus, sitting in the chest, this layer is instead called the adventitia. A favorite memory trick is my sister makes sandwiches for mucosa, submucosa, muscularis, and serosa. Surrounding these organs is the peritoneum, a double membrane lining the abdominal cavity. Its fluid lets organs glide smoothly, and folds called mesenteries anchor the gut while carrying its vessels. Organs like the pancreas and most of the duodenum sit behind it, called retroperitoneal, so their pain often appears as back pain. What we just covered is exactly the kind of material in our free practice question bank. These are not basic recall questions either. They are designed to test your deep understanding, just like the real exam will. Working through them now will pay off enormously on test day, so do not skip them. Now let us follow the food. Digestion begins in the oral cavity, your mouth. 3 things happen here. The teeth and tongue break food apart, saliva begins chemical digestion, and the mixture is shaped into a soft ball, the bolus. The roof has a bony hard palate and a muscular soft palate. Let us talk teeth. Adults have 32 permanent teeth in 4 types. Incisors at the front are sharp chisels for cutting. Canines beside them are pointed for tearing. Premolars and molars at the back grind food into paste. The crown is covered in enamel, the hardest substance in your body, which cannot regrow. The chewing muscles are worth knowing. The masseter is the primary one, incredibly strong for its size, snapping the jaw shut with great force. The temporalis, a fan-shaped muscle on the side of the head, both closes the jaw and pulls it backward. Together they create the crushing motion of mastication. 3 pairs of salivary glands flood the mouth with about 1.5 liters of saliva daily. The parotid glands, the largest, sit near your ears and swell during mumps. The submandibular glands sit under the jaw, and the small sublingual glands sit under the tongue. Each drains into the mouth through ducts. Saliva is mostly water, but that small fraction is powerful. It carries mucus to lubricate the bolus, salivary amylase to start breaking down starch, an antibacterial enzyme called lysozyme, and antibodies for defense. Saliva also dissolves food, so your taste buds can detect it. Once the bolus hits stomach acid, salivary amylase switches off. Once the bolus is ready, the tongue pushes it into the pharynx, the shared passage for food and air. Swallowing, properly called deglutition, happens in 3 phases. The first is voluntary, as the tongue pushes backward. The next 2 are automatic reflexes. Over 25 muscles fire in sequence, taking only a few seconds. Here, safety matters most. As you swallow, the soft palate seals off the nose, and a cartilage flap called the epiglottis folds down over your airway. This forces the bolus into the esophagus rather than the windpipe. Breathing briefly stops too. When this reflex fails, food goes down wrong and you choke. And from there, the bolus enters the esophagus, a muscular tube about 25 centimeters long that simply delivers food to the stomach, doing no digesting. Peristaltic waves push the bolus down in seconds, so effective you could swallow upside down. At the bottom, the lower esophageal sphincter relaxes to let it into the stomach. I know we are covering a lot of detailed anatomy, and you might wonder how to practice it. That is why I built those free practice questions. They help you work through problems combining several concepts at once, exactly how this exam challenges you. The link is in the description. Now we reach the stomach, a J-shaped muscular sac in the upper left abdomen. Its 2 jobs are churning food into a soupy liquid called chyme, and beginning the chemical digestion of protein. A normal stomach holds about 1.5 liters, but can stretch to 4 liters when full. The stomach has 4 regions exams often ask about. The cardiac region surrounds the entrance from the esophagus and receives the bolus. The fundus is the dome bulging above it. The body is the large central mixing chamber, and the pyloric region funnels down to the pyloric sphincter, the gateway into the small intestine. When empty, the stomach lining folds into ridges called rugae. These let it expand as you eat and add surface area for secretion. The 3 muscle layers churn vigorously, mixing food with gastric juice. Chyme is squirted toward the pylorus, and anything too chunky is pushed back for more mixing, a process called retropulsion. Tiny pits in the lining lead to gastric glands, and their cells are a top exam topic. First, parietal cells secrete 2 things. One is hydrochloric acid, which makes the stomach intensely acidic, kills bacteria, and unfolds proteins. The other is intrinsic factor, needed to absorb vitamin B12 in the ileum. Next are the chief cells, which secrete pepsinogen, an inactive protein digesting enzyme. Acid from parietal cells converts pepsinogen into active pepsine. Pepsine then chops dietary proteins into shorter chains called polypeptides. This is a beautiful safety design since the enzyme is stored inactive, so it cannot digest the very cells that make it. The third key cell is the G cell, which releases gastrin into the blood. Gastrin conducts the stomach. It tells parietal cells to make more acid, chief cells more pepsinogen, and muscle to contract harder. Food triggers gastrin. Gastrin boosts digestion, and that digestion triggers more gastrin, a positive feedback loop. 2 more cell groups complete the picture. Other enteroendocrine cells release helpers like histamine, which strongly drives acid, serotonin, which boosts contractions, and somatostatin, which acts as a brake. Meanwhile, surface mucus cells coat the lining with thick bicarbonate-rich mucus. Without that protective blanket, the stomach would digest itself, producing painful peptic ulcers. How fast does the stomach empty? A typical meal takes 2 to 6 hours. Liquids leave quickly, carbohydrate meals empty fastest, and fatty meals slowest, since fat is a powerful brake. The pyloric sphincter opens just a little at a time, releasing small squirts so the small intestine is never overwhelmed by incoming acid. For your exam, lock in these pairings. Parietal pumps protons, meaning parietal cells make acid. Chief chews protein through pepsin, and G cells give gastrin. A common trap is mixing up parietal and chief cells, so repeat those phrases until automatic. Also, destroying parietal cells removes intrinsic factor, causing a condition called pernicious anemia. If these stomach details feel like a lot right now, do not worry. That is completely normal. It is exactly why I created that comprehensive practice question bank. Working through those problems turns shaky recognition into confident recall. Grab the free link in the description and test yourself on these cell types tonight. Now for the workhorse, the small intestine, roughly 6 meters long, where digestion finishes and most absorption happens. It has 3 parts in order. The duodenum comes first and is shortest. The jejunum is the middle where most absorption occurs, and the ileum is the longest final stretch. Remember the phrase, dude just is. The duodenum is the busiest chemical hub of digestion. Curving around the pancreas head, it receives 3 streams: acidic chyme from the stomach, bile from the gallbladder, and enzyme-rich juice from the pancreas. The bile and pancreatic ducts usually join, guarded by a muscular valve called the sphincter of Oddi. To absorb efficiently, the small intestine maximizes surface area with 3 nested folds. First, large circular folds make chyme spiral slowly through. Second, finger-like projections called villi cover them. Third, each villus cell has thousands of microscopic microvilli forming a brush border. Together, these give a surface area near 200 square meters. Each villus is beautifully equipped. Inside sits a network of capillaries to absorb sugars and amino acids, plus a tiny lymphatic vessel called a lacteal to absorb fats. The brush border holds enzymes that finish digestion at the surface. Maltase, sucrose, and lactase split sugars. People lacking lactase suffer the familiar lactose intolerance. Now meet the liver, the largest gland in your body, mostly in the upper right abdomen. Among its many jobs, the digestive one is making bile, a yellow-green fluid containing bile salts derived from cholesterol. Here is a point examiners love. Bile contains no enzymes. Instead, it emulsifies fat into tiny droplets. Why does emulsification matter? Breaking one big fat droplet into many small ones hugely increases surface area, so the real fat enzyme, pancreatic lipase, works far faster. Beyond bile, the liver detoxifies alcohol and drugs, converts toxic ammonia into urea, stores glycogen and vitamins, and makes vital proteins like albumin and clotting factors. The gallbladder is a small green pouch under the liver, and its only job is storing and concentrating bile between meals. When fatty chyme enters the duodenum, the hormone CCK signals the gallbladder to squeeze. Bile flows down the ducts, the sphincter of Oddi relaxes, and bile pours in to tackle fats. The pancreas sits behind the stomach and is a double agent. Its exocrine part, about 99%, makes pancreatic juice full of enzymes. Pancreatic amylase finishes carbohydrates. Pancreatic lipase is the main fat digester, and trypsin leads protein digestion. Trypsin arrives inactive, switched on in the duodenum, then activates other proteases. Pancreatic juice also carries bicarbonate, a base that neutralizes stomach acid, so intestinal enzymes work in a near-neutral environment. The other 1% of the pancreas is endocrine. Its islet cells release hormones. Beta cells make insulin to lower blood sugar. Alpha cells make glucagon to raise it. Damaged beta cells cause diabetes. Let us see where absorbed nutrients go. Sugars, amino acids, vitamins, and minerals enter the villi capillaries. That blood drains first into the liver through the hepatic portal vein, giving the liver first pass at everything. Fats are the exception. Packaged into chylomicrons, they enter the lacteals and travel through the lymph. Let us pull the enzymes together simply. Every digestive enzyme fits one of 3 families. Amylases break carbohydrates, lipases break fats, and proteases break proteins. Carbohydrates are handled by salivary and pancreatic amylase plus brush border enzymes. Proteins by pepsin, then trypsin and friends. Fats wait for bile and pancreatic lipase. Chemically, digestion is wonderfully simple. We eat large polymers and must reduce them to small monomers we can absorb. Starch breaks into glucose, proteins into amino acids, and fats into fatty acids and monoglycerides. Each bond is split by adding water, a reaction called hydrolysis. That single idea unifies the whole chemistry. Remember that mastering these enzymes and organs takes repetition, not just listening once. The free practice questions are built specifically to turn what you're learning now into exam-ready skill. Each question mirrors the real format, so practicing them is the closest thing to sitting the actual test. Find the link in the description. After the small intestine, leftovers enter the large intestine. about 1.5 meters long and wider. Its main job is absorbing remaining water and salts, turning liquid residue into semi-solid feces. It begins at the cecum, where the appendix dangles, then becomes the colon framing the abdomen. The colon travels a clear path you should memorize. From the cecum it rises as the ascending colon, crosses as the transverse colon, drops as the descending colon, curves through the sigmoid colon, and empties into the rectum. A handy phrase is Carl ate 10 donuts for cecum, ascending, transverse, descending, and sigmoid colon. The large intestine looks different too. It has no villi, since little nutrient absorption happens here, but plenty of mucus-producing goblet cells to lubricate feces. It also hosts trillions of bacteria. These microbes ferment fiber we cannot digest and even manufacture vitamin K and several B vitamins which we absorb. Movement here is slower and gentler. Haustral contractions gently mix contents for water absorption, while stronger mass movements push everything toward the rectum a few times daily, often after a meal. Balance matters greatly. If contents move too fast, too little water is absorbed and diarrhea results. Too slow and excess removal causes constipation. Finally comes defecation. As feces stretch the rectal wall, receptors trigger the defecation reflex. The internal anal sphincter, which is involuntary, relaxes automatically, but the external anal sphincter is under your voluntary control, letting you decide if the moment is appropriate. When you relax it and bear down, the waste is expelled, completing the journey. Now let us tie it together with control. Because digestion is carefully regulated by nerves and hormones, nervous control comes from two opposing branches. The parasympathetic system, called rest and digest, ramps digestion up, increasing secretion and motility. The sympathetic system, fight or flight, shuts digestion down to send resources to your muscles instead. The main parasympathetic nerve to the gut is the vagus nerve, cranial nerve 10, so connect those whenever you see them. Remarkably, the gut has its own nervous system, the enteric nervous system, with over 100 million neurons. Nicknamed the second brain, it runs many reflexes alone, so digestion continues even after spinal injury. Hormonal control uses 3-star hormones you must know cold. First is gastrin, released by G cells when food arrives. Gastrin gets the gastric glands going, boosting acid, pepsinogen, and contractions. Think of gastrin as the stomach saying, food is here, let us get to work, and ramping up everything needed to digest a meal. The second hormone is secretin, released by the duodenum when acidic chyme arrives. Secretin tells the pancreas to release watery bicarbonate, neutralizing that acid so intestinal enzymes survive. A simple aid is secretin secretes bicarbonate. It also slows the stomach. Without this bicarbonate, the acidic chyme would damage the delicate intestinal wall and stall digestion. The third hormone is cholecystokinin, or CCK, released by the duodenum when fatty, protein-rich chyme arrives. CCK does 3 things. It tells the gallbladder to contract and release bile, tells the pancreas to release enzymes, and slows the stomach. Remember, CCK contracts the gallbladder, calms the stomach, and cranks up the pancreas. All this comes together in the 3 phases of gastric control. The first is the cephalic phase, triggered by the sight, smell, or even thought of food. Acting through the vagus nerve, your brain primes the stomach before a single bite arrives, increasing acid and enzyme secretion so it is ready and waiting. The second is the gastric phase, beginning when food enters the stomach. Wall stretching and proteins trigger G cells to pour out gastrin. This drives the biggest share of acid secretion through that positive feedback loop we discussed. The gastric phase does most of the heavy lifting during a meal. The third is the intestinal phase, beginning as calm enters the duodenum. At first, it mildly encourages the stomach, but as fatty acidic calm accumulates, it flips to strong inhibition. Through secretin, CCK, and the enterogastric reflex, it slows the stomach, protecting the small intestine from being flooded faster than it can safely process. We are nearing the finish, the perfect time to cement everything with practice. Those free questions cover every topic from today, from cell types to hormones to the 3 phases. They are completely free and designed specifically for exam success. So click the link in the description and start practicing right away. Let us bring it home. You now understand the digestive system as one connected journey. Food is ingested, digested, absorbed, and finally eliminated. It travels through the alimentary canal, while the accessory organs, the salivary glands, liver, gallbladder, and pancreas add their secretions. Every region has specialized cells and structure suited to its task. See how the pieces connect. The stomach acid activates pepsin and demands protective mucus. The acidic chyme it produces triggers secretin, calling for pancreatic bicarbonate. Fatty chyme triggers CCK, summoning bile and pancreatic enzymes together. Nerves and hormones constantly fine-tune the timing. Nothing here works alone, and that integration is exactly what examiners test. So how should you study? Lean on the memory phrases, master the cell and hormone pairings, and trace food from mouth to anus without hesitation. Understanding why each step happens carries you further than rote memorizing ever could. You have built a genuinely strong foundation today, so feel proud and confident heading into your exam. Now put this knowledge to work. Do not forget to grab those free practice questions from the link in the description. With the deep understanding you gained today, plus focused practice, you will be incredibly well prepared. Walk in calm and ready and go earn that passing score. You have got this.